Good afternoon, everyone. So the first year of bachelor at the University of Brussels for me was very interesting because I wanted to become a scientist and we were studying mathematics, physics, chemistry, and biology. The second year was more difficult because I was equally fascinated by physics and biology, but I had to choose. I had to choose to become a mathematician or to become a physicist or to become a chemist or to become a biologist. So eventually I chose to become a biologist. At the end of the master, I started a PhD at Yale University and I became even more specialized. And at the end of my PhD, I was a molecular phylogeneticist, a very, very specialized scientist, meaning that I was using DNA and protein sequences for reconstructing the phylogeny of life. But then, after developing a group of research in that field, I decided many years later at the University of Geneva to shift my research interest towards combining again biology, physics, and computer science for a better understanding of evolution and development of animals. So when you look at the classical curriculum, you have to make choices all the time. And the average student at the end of his or her studies doesn't realize that there is a huge overlap among disciplines. In fact, if you look at some specific subfields of biology, we are using methods and concepts that are very distant from those used in other parts of biology, but are very close to those used in some parts of physics. So, of course, there are choices to be made because you cannot study everything. But I think that there are not enough bridges in between disciplines, such that people, and I mean the average student, doesn't realize at the end of his or her studies the huge overlap among disciplines. So I will give you two examples of research topics that have been going on in my laboratory at the University of Geneva, really at the interface between biology and physics. I will speak about chameleons and how they change color, and then I will also speak about crocodiles. Let me start with the crocodiles, and let's discuss how the crocodile got its head scales. The initial observation is this one. When you look at a snake, for example, you have a very stereotyped organization of the scales. This is the reason why morphologists gave them names. You have the labial scales, you have the nasal scales, the internasal scales, etc. Now, when you look at a crocodile, it looks much more messy. And this is actually why morphologists didn't give any names to these scales, because it's highly variable from individual to individual. So in my lab, we decided to analyze that quantitatively. So we scan with this robotic system, we scan the animal under anesthesia, and we can reconstruct the 3D geometry and the color texture of the animal. And as you can see in this video, you end up with both the 3D geometry and the texture, the color texture. And if you zoom into the model, you can see all these different spots that correspond to sensory organs, but more importantly, for what I'm going to tell you today, we can mark these scales as a network of edges and nodes. We can then identify what we call the sagittal plane, which is separating the left and the right, and we can make a mirror image of the left to the right and right to the left. And when you do this, you realize indeed that there is no correspondence whatsoever between the left and the right. This is what you see in the right part of the slide, and if you do the same exercise on a snake, as I discussed previously, you can realize that there is a very good correspondence between left and right in the snake. So why this difference between crocodiles and other reptiles? In fact, by analyzing these networks of scales on the face and jaws of crocodiles, we realized that statistical signatures of what we observed on these animals were identical to the statistical signatures that we find in cracking, physical cracking, like, for example, when mud is cracking when it's drying, or when porcelain is cracking when it's cooling. You have a pattern of cracks that have some specific features that we do find in the crocodiles. I'm not going to go into the details of the physics aspects of it, but again, we can recognize a cracking pattern from another kind of pattern. And in the crocs, we do find these signatures like incomplete edges, we find many reorienting edges, and we find many 90 degrees angle connections, which is very typical of cracking systems, especially at the border of the network. Now, of course, this was an analysis performed on juvenile crocodiles, and the next step was to check into the embryo, of course, what was going on. So let's go back for a minute to the snake. Here is an embryo of a snake in which we labeled with methods I will not discuss in details, we labeled group of cells that will form scales. So each of the spots you see here on the body of the animal is a group of cells that will collectively develop a scale. So you can appreciate the existence of the pattern before the scales develop. Now, each of these spots is therefore a developmental units that is genetically determined. And you can see them on the face of the animals as well. You can already recognize the group of cells that will develop the labial scales, for example. So what is happening in the crocodile? We find exactly the same process going on the body. As you can appreciate here, we indeed have these developmental units that are forming on the body of the crocodiles, but not on the face. On the face, you do see developmental units, each of these spots, but these do not correspond to scales. They correspond to these sensory organs I was telling you before. In fact, what we observe is that the face is first smooth, and then we have grooves, crack, that appear and that propagate first across the face of the animal, and then later in development, you have new cracks that appear in other directions, such that at the end of development, what is called a scale on the face of a crocodile is not a scale developmentally speaking. It is actually a random piece of skin that has the shape and the size that it has just because of the random connection of the different cracks that propagate it. So next time you see cracked pavement, for example, you're obviously looking at a physical process, but actually the spatial organization of scales on the face and jaws of crocodiles is generated through a physical process that is extremely similar to the one you observe on cracked pavement. Now, let me take the second example about the colors of reptiles, and I'm going to discuss how chameleons change color. First, let me remind you that, of course, light is an electromagnetic radiation, and the visible range of colors is, of course, a tiny portion of this light spectrum that is going from gamma rays to radio waves. So the visible range is between 400 and 700 nanometers. And let's ask ourselves the question, why is this lizard green? Because there is no green pigment in lizards. In fact, it is green for the reason that there is a mixture of two components. The first component is a yellow pigment. And it is yellow because it's absorbing blue wavelengths. So if you remove the blue wavelengths, you are left with all the other colors, and the mix of these colors give the impression of yellow. And sure enough, if you remove the pigment with chemical treatments, you transform the green skin into a skin that is much more blue. So the question, of course, is what is this blue component? This blue component is not due to the presence of a pigment. It is due to the presence of cells under the yellow pigments, and these cells are very special. They are called Iridophores, and they contain tiny nanocrystals of guanine. And light waves will interact with this iridophore and produce a complex physical phenomenon which is called light interference. The net effect is that the cell will act as a selective mirror. All light will go through except some specific wavelengths, and in this case, it will be the blue and the green wavelengths that will be reflected with 100% efficiency. It is the same phenomenon that you can observe on soap bubbles, for example. Although there is no pigment in the soap, you observe the presence of colors on the soap bubble. This is also due to light interference. In the iridophores, it's just much more efficient. Now, the final point I want to raise here is that the selection of wavelength will be a function of the distance among the stacks of guanine. Now, I could stop here and tell you that this lizard is green because of these two components. You have the yellow pigment and you have the blue light which is reflected. So when you have blue and yellow and you mix them, you get green. But that would not be totally accurate. What is happening actually is that the white light of the sun contains all these different wavelengths, and the red and yellow wavelengths are just going through the skin. So you have the skin here with the yellow pigment and the structural elements, and you have the muscles below the skin. So the yellow and the red go through because it's not absorbed by the pigment and it's not reflected by the photonic crystal. The blue component is absorbed by the pigment on the way in, and the blue light that was not absorbed yet here will be absorbed on the way out of the skin. So the only light that remains is the green light. The green light is not absorbed by the pigment and is reflected by the photonic crystal, so the animal looks green. Now, I didn't tell you yet how the chameleon is changing color. They are changing color in two situations, mostly during male-male combat, or when a mature male is trying to impress a female and trying to attract the female in the tree. So this is a display that the animal is doing. It's going from a cryptic coloration to a very visible coloration. This is basically communication. Now, for many years, people have thought that chameleons were changing color through dispersion aggregation of pigments in their skin. And this was not a bad hypothesis. It was based on the fact that many lizards, including chameleons, can indeed disperse and aggregate melanin in these very big cells that are called melanocytes. They have digitations that allow the pigments to be dispersed or to be concentrated in the cell body. So, of course, the animal can become dark or can become bright. But that does not explain the change of color from green to yellow. In fact, we found in the skin of chameleons, we found these iridophores that are very special because they contain nanocrystals that are extremely well organized. So these are two sections in different orientations of the skin. And in this orientation, you can appreciate the squared organization. And here you have a little hexagon. And these are really tiny crystals. You have a scale here of 200 nanometers. So the first thing we did to analyze the process was to simply film with higher resolution the animal while it was changing color. We took each frame of the video and we computed the overall color of the animal on each of these frames, and we plotted this value on the color space that you see here. And as you can see on the graph, the animal is losing a blue component that is transformed into a green and then a yellow and red component, which explains that the animal is shifting from a cryptic green to a very visible yellow or orange. Now, we came up with this surprising hypothesis that maybe these animals were doing this through modification of the lattice parameter of their iridophores. Maybe they are able to change the distance among the nanocrystals. Indeed, I was telling you that the reflection of these iridophores will specifically depend on the distance among the nanocrystals. So for example, if we would physically compress the crystals, we would get a shift of reflected wavelengths from green to blue. And that's what you see on this movie here. We are simply compressing the skin with tweezers here, and you can appreciate that the skin is changing from green to blue. So for chameleons, we performed numerical simulations with the quantum material groups at the University of Geneva, and it consists into computing the optical response of the iridophores for various distances among the nanocrystals. And the result we obtain is the white line you see on the left of the screen, and you can see that the fit between the numerical simulations and the observations is really excellent. In addition, if you observe the iridophores of the skin of an animal which is in its cryptic state, you can see that the nanocrystals are close packed. Whereas if the animal is excited, the crystals are more distant from each other. The final experiment that we did was to take a piece of skin ex vivo, put that in a petri dish under a microscope, and then to modify the amount of salt in the solution in which you keep the skin. This way, we make the cells swell or shrink, depending on the amount of salt we are putting in the solution. And here, we are just focusing on one cell, and the cell is swelling. So at the beginning, it was reflecting blue, then it's reflecting green, then it's reflecting red. And you can, of course, go in the other direction. You can make the cell shrink, and indeed, you are going from red to blue. And the fit, again, with the numerical simulations is very good. So the two examples are discussed here, how the crocodiles got its head scales and how chameleons change color. I don't think that we would have been able to discover these mechanisms if we were not on a daily basis in the laboratory combining physics and biology. And I want to stress for students of science that, of course, they have to make choices during their studies, but they have to be aware of this huge overlap among disciplines, and for example, between biology and physics. And I'm pretty happy to see that today biophysics is really a booming field, but there is more than just biophysics, of course. And I think that we should be very flexible in the curricula, and we should train a new generation of chameleon scientists who are at the interface of different disciplines, because that's where I really think that many of the most interesting research topics are. Thank you for your attention.